Welcome to this lecture series on eukaryotic gene expression, basics and benefits. The last 3 or 4 lectures we have been discussing about the various eukaryotic expression systems. We began by discussing how genes can be cloned and expressed in yeast and insect cells. Then we moved on to mammalian cells, and in the last class we discussed about how you can introduce genes and express them using non-viral vectors. And today we are going to discuss about expressing genes in mammalian cells using viruses as gene delivery vehicles or gene transfer vehicles or viral vectors. So this is the summary of what we have been discussing in the last few lectures. We discussed in the lecture number 30 about protein expression in yeast and insect cells. Then we discussed about how non-viral vectors can be used for transfecting genes and plasmids into mammalian cells. Then we also briefly discussed about the various cell-free protein systems that are available and how we can actually make cell-free systems like rabbit reticulocyte lysates, wheat germ extracts, and more recently you can also make using the cell-free systems huge amounts of proteins, even milligrams or even gram amounts of vectors, proteins can be made using cell-free expression systems. So today the focus is going to be on how we can introduce genes and express them so that your protein of interest can be produced in mammalian cells, but our focus is going to be on how viruses can be used for introducing genes into mammalian cells and expressing them very efficiently. This lecture is very important because both this lecture and the previous lecture actually forms the basis for a very fascinating area of research which has taken off in the late 1990s, namely the human gene therapy. So unless we understand how people are trying to introduce genes into mammalian cells using non-viral vectors and viral vectors, we will not be able to understand some of the important advances and very exciting developments that are taking place in the area of human gene therapy. So let's spend some time to understand how viruses are being used for delivering genes into mammalian cells and how we can express your gene of interest so that the proteins can be made in mammalian cells. So in the last class, we basically discussed about a number of non-viral techniques as well as non-viral vectors, basically plasmid-based vectors, using either viral promoters or certain cellular promoters for expressing your gene of interest. And we discussed a variety of methods like the calcium phosphate precipitation, using cationic lipids and liposomes, electroporation. There are also many other methods like DA dextrans, directly injecting DNA, and some of these methods we'll discuss much later in this lecture series. Basically what we discussed is that a number of transfection protocols have been established to introduce the expression plasmid of your interest into mammalian cells. And these are all termed together as non-viral gene delivery techniques. Now, why do you have to now discuss about using viruses as gene delivery vehicles? What is wrong with non-viral gene delivery? The two major problems with non-viral gene delivery mechanisms are, one is the efficiency of transfection. When you use reagents like liposomes or even electroporation or even calcium phosphate precipitation, the percentage of cells into which you can introduce your plasmid or expression plasmid is rather low. So compared to viruses for gene delivery, the non-viral vectors are not as efficient as the viral vectors as far as the efficiency of transfection is concerned. So if you really want to express a huge vast majority of cells with your gene of your interest and produce large amounts of this protein, the non-viral gene delivery techniques poses certain problems. The other major problem with the non-viral gene delivery mechanism is that the expression plasmid that I have introduced into the cells, you are only introducing into the cytoplasm, but now you have to take them into the nucleus. So unless your expression vector goes inside the nucleus, your gene will not be transcribed and RNA will not be translated into your protein of interest cytoplasm. So there are a number of the nuclear envelope actually serves as a major barrier for successful non-viral transfection of exogenous genes as well as their subsequent expression both in vitro and in vivo. And the nuclear envelope poses a major problem for transfections in which the exogenous DNA is delivered into the cytoplasm. So after the delivery into the cytoplasm, the expression vector of the nucleic acid becomes complex with cellular proteins that mediate interactions with cellular machinery for trafficking into the in and out of the nucleus. In fact, there is a very nice review in gene therapy about the progress and prospects nuclear import of non-viral vectors, where you actually discuss a number of problems associated with delivering your vector into the nucleus. But today, in fact, we have mechanisms or we have devices and methodologies for actually introducing your DNA or your nucleic acids right into the nucleus. These are all called as nucleofection techniques. There are apparatuses which you can buy, and you can actually introduce your vector right into the nucleus using these nucleofection vectors. So the non-viral vectors have now gained some momentum with technologies such as nucleofection techniques. But with some of these limitations, people started wondering how we can introduce genes more efficiently into a vast majority of cells. And one thing that came into the mind is, why can't we use viruses as gene delivery vehicles? The reason is, a number of viruses belonging to a variety of families of viruses, variety of classes of viruses like papaviruses, papillomaviruses, parvoviruses, adenoviruses, herpes viruses, retroviruses, lentiviruses, they all infect humans and animals. And these viruses have been infecting us from time immemorial, and therefore they have also devised a variety of mechanisms by which they can attach to cells, push their genomes inside the mammalian cells, and the viral genome can efficiently transcribe their genes, and also they can replicate and they can come out of the cells, either lysing or without lysing. So since the viruses have devised natural mechanisms, they can infect cells, mammalian cells. Well, people thought we use these viruses as natural gene delivery mechanisms or gene delivery vectors. So since viruses have evolved specialized molecular mechanisms for efficiently transporting their genomes inside the cells they infect, people started asking the question, why can't we now modify these viruses in such a way that instead of they going and infecting and replicating their genome and transcribing their genes and producing their viral proteins, can we now introduce our gene of your interest into a virus, so that when the virus goes and infects the mammalian cell, instead of making the viral proteins, it now makes your protein of your interest. So this concept of using viruses as gene delivery mechanisms gained a huge popularity, and in fact today some of the most efficient mammalian expression system that we have is based on viral vectors. So let's now try to understand how viruses can be used for delivering your genes into mammalian cells, and how you can express efficiently once the gene is introduced into the mammalian cell. So the focus is going to be on how do you construct a viral vector. So you have a viral genome which codes for a number of viral proteins. Now we have to take out some of the viral genes and put your gene of your interest, and this constitutes what is called as a construction of a viral vector. Now once you have a viral vector, from this you have to generate what is called as a recombinant virus, so that this virus can now go and efficiently infect your cell of your interest. So there are two steps in using viruses as gene transfer vehicle or virus mediated gene delivery into mammalian cells. The first step is construction of a viral vector, where you can introduce your gene of your interest, and once you have a recombinant viral vector ready, from that you have to generate a recombinant virus. How do you achieve this? The general strategy that is employed for using viruses as gene delivery vehicles is schematically shown here now. Now let's take a normal viral genome. It can be a retrovirus or an adenovirus or an adeno-associated virus. This is a very general simplified scheme which is applicable to all kinds of virus mediated gene transfer mechanisms. Usually, normally any viral genome will contain promoters and enhancers, and through which transcription factors, usually the host transcription factors, go and bind to these promoters and enhancers, and they transcribe a number of viral genes which are downstream. I have designated here some of the genes as P1, P2, P3, P4, etc. So usually the viral genomes will have regulatory elements that is necessary for transcription of the viral genes, and also at the 3' end you will also have something like polymorphisation signal, processing of the RNA, and so on and so forth. So the general strategy of now converting this virus genome into a viral vector is, you now delete some of the viral proteins which probably are not very essential, or which are called as structural genes, which actually code for structural components of the virus, like the capsid, the envelope, and so on and so forth. Delete those genes. In this case, for example, I have shown that I have deleted P1 and P2 from this viral genome, and clone your gene of your interest in the place of a P1 and P2. And if you now have a backbone in which it has a E. coli origin of replication and a selection marker such as an antibiotic marker, you can now, once you ligate your gene of your interest into a plasmid, you can easily replicate this plasmid in large amounts in E. coli. So you will now have generated a recombinant viral vector. The first step has been achieved now. So by removing certain viral genes and introducing your gene of your interest, you have now generated a recombinant viral vector. Now from this, how do you generate a recombinant virus? what you necessarily do is, you now take this gene and then introduce into a cell line called as packaging cells. What are packaging cells? Packaging cell lines are nothing but cell lines that package the recombinant retroviral, so it can be retroviral or it can be any viral RNAs or viral genomes, into infectious replication incompetent particles. Let's now try to understand what these are. What you normally do is, I told you, in generating a recombinant viral vector, you have actually deleted certain viral genes, in this case P1 and P2. So what you do is, you now take any cell line, any mammalian cell line, and clone this P1 gene and P2 gene under a mammalian expression vector and generate a stably transfected cell line which now constitutively expresses these viral proteins P1 and P2. This is called as a viral packaging cell line. That means here you have a mammalian cell line which is constitutively expressing viral proteins such as P1 and P2. Now into such a cell line, if you now introduce your recombinant viral vector which lacks the P1 and P2 but in their place contains your genes of your interest, I will designate as GOI1 and GOI2, you can put 1 or 2 genes depending upon your need. So once you transfect your plasmid recombinant viral vector into this packaging cell line, what happens? This recombinant viral genome will now be packaged into virus particles, infectious virus particles, because the viral proteins necessary for the viral packaging is being produced by this cell line. So what comes out of this cell line is actually a recombinant virus, but there is something very different from this recombinant virus compared to the wild type virus. When you infect this genome into cells, you get a recombinant replication competent virus because it contains all the viral proteins required not only for infection but also for replication of the virus. But when you transfect this particular viral genome in which you have deleted certain viral genes and put your genes of interest, when the recombinant virus that is coming out of such cells, they are known as replication defective recombinant viruses. That's because these viruses which are emerging out of these packaging cells, they contain a recombinant genome, and this recombinant genome lacks certain viral genes which are necessary for viral replication. So the viruses that emerge out of a packaging cell line following the transfection of a recombinant retroviral vector are termed as replication defective recombinant viruses. Now what do you mean by this? When you now take these viruses, infect another cell line, these viruses can go and infect because these cells contain usually specific receptors through which the viral surface proteins interact, and once the virus infects, the viral genome goes inside the cells, but instead of making the viral proteins, now your gene of your interest will be transcribed in the nucleus and the protein of your interest will be made. But since no viral proteins are being made inside the cells, a virus cannot emerge out of this infected cell. Therefore, the virus cannot replicate and an infectious viral particle cannot come out. So this is like Abhimanyu getting into a Chakravyuha. He can only go inside but cannot come out. So the virus can infect efficiently the mammalian cells and push the genome inside, and the gene can be transcribed efficiently, but a virus cannot emerge out of the cell because the viral genes necessary for successful multiplication of the virus and producing the viral progeny are missing in these replication defective viral particles. So by this strategy, you can actually generate a huge amount of these replication defective recombinant viruses, and if you now simply add these viruses to any cell line of your interest, the virus will infect those cells, your protein of your interest will be made in large amounts, but no virus will come out of these cell lines. So you are very safe. So this is a general strategy by which viruses can be used as gene transfer vehicles for expressing genes of your interest in mammalian cell lines in large amounts. I hope the concept is clear now. So basically, when the recombinant replication defective recombinant viruses infect these mammalian cells, the genome is pushed inside, the transgene will be expressed, and your protein of your interest will be made, but no virus will come out of the cell because in this case proteins such as P1 and P2 are missing, and without that virus cannot successfully replicate and recombinant viruses cannot come out of these cells. So having understood the general concept by which one generates recombinant viruses, so basically you first clone your gene of your interest into a retroviral vector in which certain genes are missing, and once you get a recombinant viral vector, you produce this virus in large amounts by transforming this plasmid into E. coli. And once you get this plasmid in large amounts, you take this plasmid, transfect them into a mammalian cell line, which is actually called as a packaging cell line, because this cell line is actually producing certain viral proteins that can package your recombinant viral genome into infectious viral particles. But the difference between this infectious virus particle and the normal virus infection is that these virus particles are called as replication defective viral particles, because they can only infect but they cannot replicate inside the mammalian cells. So this is the basic strategy by which one can use viruses as gene delivery vehicles, and one can generate recombinant replication defective recombinant viral particles. So using this strategy, let's now try to understand how people have been generating a number of different viral vector recombinant viruses for transfecting mammalian cells and expressing your genes of interest in large amounts. So there are two steps in using the recombinant viruses for expressing genes in mammalian cells. One is the generation of a retroviral vector from a viral genome, and once you have a viral vector, clone your gene of interest, and then you can actually now convert a normal virus, infectious replication competent virus, into a replication defective recombinant virus. These are the two major aspects by which viruses can be used as gene delivery vehicles. So what I'm going to do in the next few minutes is to discuss with you how people have generated recombinant adenoviral vectors and recombinant adenoviruses, recombinant adeno-associated viral vectors and recombinant adeno-associated virus, similarly recombinant retroviral vector, recombinant retrovirus, recombinant lentiviral vector, recombinant lentiviruses. There are many other types of viruses, but since time does not permit to discuss everything, we'll use these 4 different viral systems. I think I'll also briefly mention about Vaccinia vectors, how Vaccinia viruses can be used for generating recombinant Vaccinia viruses for making your proteins in large amounts in mammalian cells. So let's go one by one and see how these viral systems have been developed. Let's first discuss how gene transfer and expression has been carried out using adenoviruses and adeno-associated viruses. These are both are DNA viruses. So the adenovirus, adeno-associated virus, as well as the Vaccinia virus are the examples for DNA viruses. How DNA viruses can be used for expressing your genes of interest. The retrovirus and lentivirus are example for RNA viruses or retroviruses as a gene transfer vehicles. Now what is an adenovirus? The adenovirus is a non-enveloped viral particle. It contains a linear double-stranded DNA, and once an adenovirus infects any mammalian cell, it infects a wide variety of mammalian cells. The viral genome does not go and integrate with the host genome. It remains as an episome or an extra chromosomal DNA, and it replicates inside the host cell nucleus as a extra chromosomal DNA. So the viral DNA will be transcribed, the viral proteins will be made, and then the viral DNA will be assembled and infectious viral particles emerge out of the cell. This is just the structure of a normal adenovirus. And if you look at the adenoviral genome, it contains a number of transcription units or number of genes which are transcribed usually in 3 different phases of the virus life cycle. Genes which are transcribed immediately after the virus infection of a mammalian cell are known as immediate early genes. This is what the genes designated as E1A and E1B actually are immediate early genes. Usually these immediate early genes are nothing but transcription factors which are required for the transcription of the next phase of the viral genes, namely the early genes. So once the immediate early genes are transcribed, immediate early genes now go and bind to the promoter of the viral promoters and trigger the expression of the early genes. And these early genes then following the early gene expression, you get what is called the late genes which are transcribed, and following the expression of all these immediate early genes and late genes, finally the virus replication as well as virus transcription is over, and then the viral DNA gets assembled into the viral proteins and an infectious viral particle emerges out. So the strategy for converting such an adenoviral genome into an adenoviral vector is you basically delete the immediate early genes such as the E1A, E1B or even the E3, and in their place put your gene of interest. So this is what some of the normal very popular adenoviral vectors have been generated here. For example, you have an adenoviral vector in which the E1 gene has been deleted, so it is known as the delta E1. And what you do, if you want a recombinant adenovirus to generate from such a vector, you have to take this E1 deleted adenoviral vector and put them in a packaging cell line that expresses E1 protein. And therefore, you will get a recombinant adenovirus which is again replication defective. That is, these recombinant adenoviruses can multiply only in those cell lines which can express E1A. If the cell of mammalian cell line does not express E1, then the virus will not replicate. It can only infect and your transgene of your interest will be expressed, but the virus will not replicate. Such even deleted adenovirus vectors can actually carry about 3 to 5 kb of your transgenes. If you have a gene bigger than that, you cannot use the even deleted adenoviral vectors. So to accommodate larger fragments of DNA, people have started deleting other genes and started putting these genes in packaging cell lines so that these proteins can be provided in trans. So like that you have E1 and E4 deleted, E1 and E2 deleted, or E1 and E3 deleted, and so on and so forth. So basically you delete these genes from the adenoviral genome and clone your transgene of your interest in their place, and then transfer them into packaging cells which are now expressing those missing proteins. Therefore you get a recombinant adenovirus. The more you delete from the adenogenome, larger fragments can be cloned. So up to 10 to 9 kb of your genes can be cloned into these kinds of adenoviral vectors. So the strategy has been shown here. The human adenovirus type 5 is what is normally used for making these adenoviral expression systems. Usually you have either an E1 deleted virus or an E3 deleted virus. When you do this, they become replication incompetent. They cannot replicate in mammalian cells which do not express E1 or E3 proteins. Up to 8 to 10 kb of foreign DNA can be inserted into such kind of a replication defective adenoviral vector genomes, and usually when you take such replication defective adenovirus infected cells, they do what is called as a non-cytopathic or non-lytic infection. That is, adenoviruses don't lyse the mammalian cells, but they bud out of the mammalian cells, therefore the cell still remains alive. So the strategy usually is to generate this kind of a replication defective adenoviral vectors and transfer them to packaging cells. One of the most popular cell line which is used for generating a recombinant adenovirus is called the HEK293 cells. These HEK293 mammalian cells actually express the E1 protein of the adenovirus. So when the replication defective adenoviral vector is introduced into the cell line, you get a replication defective adenovirus emerging out of these cells. Now, today we have a number of companies which actually sell these kinds of adenoviral expression systems, so you don't really have to construct recombinant adenoviral vectors or generate packaging cell lines. You can just go and buy these expression systems from a number of commercial companies. You just take and clone your gene into those adenoviral vector systems and take the packaging cell line that they provide by the company, and you can generate your own replication defective adenoviruses. For example, one of the companies which is very popular which sells these kinds of expression system is called CloneTech, and I'm just listing some of these things. For example, this company sells an adenovirus mammalian expression system known as the recombinant adenovirus Adeno-X. The company gives a complete kit for generating a recombinant adenovirus where you actually clone your gene of interest in a shuttle vector and make the shuttle vector in large amounts in E. coli, then linearize this vector using two specific enzymes, and they also supply the entire adenoviral genome, which is called as the adenovirus X genome. You can see it's about 32.6 kb DNA, which is also digests the same enzymes as you have digested your shuttle vector, and now you ligate this and this, and you get then transfer them into the HEK293 cells which now produce E1A, and finally you get replication defective recombinant virus which expresses your gene of your interest. Kits are available. All that you have to do is to buy these expression systems and clone your gene into those vectors, generate a recombinant virus, and express whatever protein you are interested in. So the adenoviral vectors have a number of advantages. They can infect in a cell cycle independent manner. They can infect primary cells as well as transformed cell lines. They have a wide broad host tropism. They can infect many different mammalian species including humans, non-human primates, pigs, rodents, mice and rabbits. They also have a broad tissue tropism. They can infect virtually any type of human cells such as skin, muscle, bone, nerve, liver cells, etc. So once you have a recombinant viral vector, recombinant adenovirus, you can actually infect a number of mammalian species as well as a number of mammalian tissues or cells with this kind of adenoviral vectors. The advantages is you have now very good expression systems available commercially, so you can generate a very high titer of the adenovirus. They very efficiently transduce both dividing as well as non-dividing cells. It's very, very important. When you come to retroviruses, retroviruses can only infect dividing cells, they cannot infect non-dividing cells. Whereas the adenoviruses can infect both dividing as well as non-dividing cells. But there are certain disadvantages. We'll discuss some of the disadvantages in the next class when we talk about human gene therapy. So the major problem with adenoviral vector system is that it's a rather big genome, almost 30 to 35 kb genome, and many adenoviral proteins still express in trace amounts when you put these adenoviruses into the mammalian cells, whereas transporting mammalian cells is not a problem. When you take these recombinant adenoviruses infect use it for gene therapy purposes, you get into lots of problems. We'll discuss these problems in the next class when we discuss about gene therapy. But because of these problems associated with adenoviral vectors, people went on to see, can we develop simpler expression systems? And one of the expression vector systems that have been developed is called as a adeno-associated virus. Now, adeno-associated virus, or AAV, is a very simple non-pathogenic single-stranded DNA virus dependent on the helper virus, usually adenovirus, to replicate. Since it's a very small genome, it contains very few viral proteins. In order for AAV to replicate in mammalian cells, it also requires co-infection by an adenovirus, because the adenovirus actually provides some of the proteins required for the AAV replication and transcription. So, AAV can successfully replicate, and viral particles can come out of mammalian cells only when it is co-infected with adenovirus. The two major genes of the AAV genome are called as cap and rep. These are sandwiched between inverted repeats that define the beginning and end of the virus and contains a packaging sequence. So, the viral genome contains what is called a packaging sequence and codes for two major proteins called as cap and rep. The cap gene encodes viral capsid proteins, and the rep gene product is involved in viral replication and integration of the AAV virus into the host genome. The adenovirus remains extrachromosomal, remains as an episome, whereas the AAV actually goes and integrates into the host chromosome. In fact, the AAV can infect a variety of cell types, and in the presence of the rep gene product, the viral DNA can integrate very preferentially into human chromosome 19. This was one of the biggest advantages of using adeno-associated virus as a gene delivery vehicle, because when you infect a recombinant adenovirus, adeno-associated virus into a mammalian cell, in presence of the rep protein, AAV goes and integrates at a specific region in chromosome 19. It does not go and randomly integrate, it goes and integrates a specific region in the chromosome 19, which has very important advantages in the next class when we discuss about gene therapy. So, AAV is a defective virus and needs a helper virus for its replication, and so far AAV does not cause any human disease. So, this is the general strategy for generating an AAV vector. So, to produce an AAV vector, the rep and cap genes are replaced with your transgene. That is what I have shown here. You have the inverted repeats of the AAV genome, and you have the two genes coding for rep and cap. All that you have to do is introduce your gene of your interest in the place of the rep and cap. So, you have generated recombinant AAV vector. So, you take this recombinant AAV vector and again put it in a packaging cell line, which is now producing rep and cap, and along with an adenoviral protein, you can successfully generate a recombinant adeno-associated virus. The adeno-associated viral vectors have a number of advantages, because it does not have any viral genes. Almost all the viral genes have been removed. They are very safe because they do not produce any disease. They can also divide, just like adenovirus, they can also transfect non-dividing cells. Since the adeno-associated virus goes and integrates specifically at chromosome 19 in human cells, they have an advantage, because once you transfect a cell line with an adeno-associated virus, the progeny will also carry a copy of the AAV gene. So, when the cells divide, the daughter cells also carry adenoviral gene, so the daughter cells also will start expressing your gene of your interest. Whereas in the case of adenovirus, since it is an extra chromosomal, when cells divide, all the cells will not inherit the adenovirus, recombinant adenovirus genome. So, in the process you will lose out, only a small population will start expressing. The disadvantages of AAV is that since the genome is very small, the amount of transgene you can put is very small. As you can say, you cannot put more than 4.7 kb of the transgene. That is the maximum you can clone into a AAV vector. If you have a gene of your interest bigger than 5 kb, you cannot use AAV as your vector of your choice for introducing genes. Producing AAV is more labor intensive, and still the AAV mechanism of life cycle of AAV is not very well understood. So, having discussed the adenovirus and adeno-associated virus, let us spend some time to see another very important DNA virus, Vaccinia virus, how Vaccinia virus has been used for expressing genes in mammalian cells. How do you generate a recombinant Vaccinia virus expression vector and recombinant Vaccinia virus as well. Now, what is a Vaccinia virus? Vaccinia virus is a huge virus. It has a genome of almost 1,87,000 base pairs. The large size of the Vaccinia virus genome prevents the construction of recombinant genomes by restriction nucleus cleavage and religation of the pharyngeal DNA. Since the retroviral genomes are all genomes are very small, but the Vaccinia virus genome is too huge, so you cannot take this DNA, simply cut some region of the viral genome and put your gene of your interest. You need to have a different mechanism. So a two-step strategy has been utilized for generating recombinant Vaccinia viruses. In the first step, a plasmid is constructed that contains a foreign gene linked to a Vaccinia virus promoter. And in the second step, this chimeric gene is inverted into Vaccinia virus genome by homologous recombination between the virus and the plasmid DNA in vivo. Let's now try to understand through a schematic representation how actually you generate a recombinant Vaccinia virus. So what you do here, first step in generating a recombinant Vaccinia virus is you clone your gene of your interest on either side of a thymidine kinase gene. This thymidine kinase gene is actually present in a Vaccinia virus which is normal. So the normal wild type Vaccinia virus contains the thymidine kinase gene. Now you generate a bacterial plasmid in which this thymidine kinase gene is disrupted by your gene of your interest. Now you take cells which are expressing Vaccinia viruses, that is, you take a mammalian cell line of your interest, transfect this plasmid and also infect the wild type Vaccinia virus. What happens? Because of the homologous recombination between the thymidine kinase regions in the vector and the thymidine kinase gene present on the chromosome of the Vaccinia virus, homologous recombination takes place and you now generate a virus which now contains a thymidine kinase gene disrupted by your gene of your interest. When thymidine kinase is disrupted, then such cells which do not express thymidine kinase gene and therefore they are resistant to a compound called BUDR or compound called ganciclovir. They can survive. Whereas the wild type Vaccinia viruses which are expressing the thymidine kinase gene, they become sensitive to compounds like ganciclovir or BUDR. Therefore, all the Vaccinia viruses emerging out of the cell line, because they produce thymidine kinase, they all die. Whereas the recombinant viruses in which the thymidine kinase is disrupted, because they cannot produce thymidine kinase, they can efficiently grow on a medium cells containing these kind of a selection markers, and therefore you can selectively grow only recombinant adenoviruses in which the thymidine kinase is disrupted, whereas the wild type viruses emerging out of the cells in which the thymidine kinase is inactive, they all die. So you can thus generate recombinant Vaccinia viruses. One of the major advances that took place in the Vaccinia expression system is generation of what are called as a non-replicating Vaccinia virus, especially particular strain called as a modified Vaccinia ankara or MVA. The problem with Vaccinia is that many Vaccinia viruses produce a number of diseases like smallpox, canary pox and so on and so forth, fowl pox. They are all known to produce number of diseases in humans and animals. So what was required actually to use these viruses safely in human cells and human systems is to generate a virus that is defective and does not cause disease. What happened? The MVA strain was actually discovered, which is nothing but it is derived from the wild type Vaccinia virus strain ankara by over 570 serial passages of the wild type virus in chicken embryo fibroblasts. So if we kept on passaging this virus in chicken embryo fibroblasts, over a period of time it lost certain regions of the genome, especially about 31 kilobases of the DNA got lost from this Vaccinia virus genome. Therefore you got a new strain of virus called as the MVA, in which because of the loss of this region of the genome, it became non-virulent or avirulent. So the MVA strain lost the capacity to productively infect mammalian cells and had 6 major deletions of DNA totaling about 31,000 base pairs, including 2 host strain genes. So these MVA vectors do not efficiently infect mammalian cells. As a result, MVA is severely host restricted. It grows well in avian cells but not in human and most other mammalian cells. So this generation of this MVA vector by Bernie Moss lab in National Institute of Health in the United States made a huge difference, and a number of laboratories now started using this MVA strain of Vaccinia for transfecting mammalian cells and using this as a recombinant viruses for generating Vaccinia based expression systems. Now I will not go into more details of the Vaccinia based expression systems. We have another lecture called as vaccines and recombinant vaccines, and in those lecture series we will discuss how these kinds of viral based vector systems are being used for producing recombinant vaccines. We will discuss vaccine in detail in that particular lecture. So I just gave you 3 examples of how DNA viruses can be used for transfection of mammalian cells and for expressing gene of interest. Each one of them has its own advantage and disadvantage. This table here actually lists some of the most commonly used DNA virus based expression systems: the adenovirus based, adeno-associated virus based. I did not discuss herpes virus based vectors, they are also being used, and also the Vaccinia, and each one of them have many advantages and many disadvantages, and one can go through this table and then see what kind of advantages and drawbacks are there in various expression systems. Let's now switch gears and see how RNA viruses can be used for as gene transfer vehicles and expressing genes of your interest in mammalian cells. We will restrict ourselves to one particular group of RNA viruses, namely the retroviruses. How retroviruses are being used as gene transfer vehicles, and how you can clone your introduce your gene into a retroviral genome, generate a recombinant repair retrovirus and infect mammalian cells so that these cells now produce your protein of your interest. The simplest form of retrovirus is called as a mononeurine leukemia virus, or MMLV. This is the one of the most successfully used retroviruses for generating retrovirus based vector systems. If you look at the genome of this MMLV, it contains what is called a 5' LTR and a 3' LTR. LTR stands for long terminal repeat. The 5' LTR actually contains the promoters and enhancers required for the transcription of the viral genes. There are 3 viral genes called GAG, POL and envelope. The 3' LTR actually contains signals for polyrelation of the viral RNA and so on and so forth for the processing of the RNA. The 3 structural genes, the GAG actually is a polyprotein, it is an acronym for group antigens. The POL is actually reverse transcriptase and envelope is the envelope protein. So the GAG is processed to matrix and other core proteins that determine the retroviral core. This forms the surface proteins of the retrovirus. The POL actually contains reverse transcriptase, RNase as well as an integrase, because retroviruses, once they infect mammalian cells, the retroviral genome goes and integrates into the host chromosome, for which you require the integrase function of the polymerase. The envelope protein actually resides in the lipid bilayer and actually determines the viral tropism. So depending upon what kind of envelope protein is being expressed on the retroviruses, some of them infect urine cells, some of them infect only human cells and so on and so forth. So the kind of envelope protein expressed on the viral surface determine what kind of cells it actually infects, or what is called as determines the tropism of the virus. So now let's see, with this kind of a simple retroviral structure, this is how the retroviral genome looks. How the retroviral, when this with the retroviral genome when infects, what is the life cycle of a retrovirus? So once the retrovirus infects a mammalian cell, this retroviral genome which I just now described now is pushed inside the cell, and transcription proceeds through the genome. The RNA, because as an RNA is the genetic material of these retroviruses, the full length message, the RNA is transcribed and the full length message is spliced related to production of the viral proteins. The unspliced full length RNA gives rise to GAG and POL proteins. The GAG, POL and POL proteins are either made as GAG protein or the GAG POL precursor, and a viral protease cleaves the precursor into multiple subunits with varying functions. The envelope protein is also translated as a precursor and then is cleaved by an endogenous protease to get a mature surface glycoprotein. And these translated proteins, once all these viral proteins are made, they assemble into a retroviral particle at the cell surface of the infected cell. The full length genomic unspliced RNA containing a packaging signal termed as Psi is bound by the GAG-derived proteins and incorporated into the budding viral particle. So this is generally the life cycle of a normal retrovirus. So once the genome is pushed inside the cell, once the retrovirus infects a mammalian cell, the genome goes inside, the RNA is reverse transcribed and gets integrated to the genome, and then the transcription proceeds, the viral proteins are synthesized, and then the unspliced viral RNA is assembled along with viral proteins and the virus buds off from the surface of the infected cell. Now let's see how this retroviral vector, as I said, the generation of a recombinant viruses has 2 steps. One is the generation of a recombinant vector and then is the generation of recombinant virus. Let's see how you can generate a recombinant retrovirus. As I said, the key components of a retroviral genome is what is called as a Psi, or the packaging signal. Without this packaging signal, the genome will not be packaged into a virion, into the viral particle, and virus particle will not come out. So this packaging signal is called Psi is very essential for the packaging of the viral genome into the virus capsid. The 3 important structural genes of a recombinant retrovirus are GAG, POL and envelope. What you do in generating a recombinant retroviral vector is, you now replace these GAG, POL and envelope genes with your gene of your interest. So you now have a recombinant retroviral vector. Again, by putting a antibiotic marker gene and E. coli origin of replication, you can actually clone this recombinant DNA into E. coli cells and make large amounts of this recombinant retroviral vector. So once you have made a large amount of recombinant retroviral vector, you then introduce this vector into a packaging cell line that expresses the virus GAG, POL and envelope genes. So the retroviral vectors provide the packaging signal psi, transcription processing elements and the target gene, that is your gene of your interest. And since the packaging cell line is now producing all the structural proteins, your viral recombinant genome will now be assembled, and from this cell line infectious retroviral particles actually can be produced. So using this kind of retroviral expression system, up to 6.5 kb genes can be cloned into these retroviral vectors, and the viral envelope gene expressed by the packaging cell line codes for the envelope protein, which determines the range of the infectivity or the tropism. This I have already mentioned. So this is how generally you generate a recombinant retrovirus. You first clone your gene of interest in the recombinant retroviral vector, in which the GAG Pol envelope proteins are not made, and once you made sufficient amount of this recombinant retroviral vector in E. coli system, you now transfect a packaging cell line which is actually producing these retroviral proteins, namely the GAG pollen envelope, and now your recombinant retroviral genome will now be packaged, RNA will be packaged inside these viral particles, and you can generate a replication defective retroviruses coming out of these packaging cells. Now one of the major concerns when they developed these recombinant retrovirus is the chance of a wild type virus emerging out of your packaging cell line. Let us see what is this problem. As I told you, you have a packaging cell line in which the 3 structural proteins of the retrovirus is being expressed from a promoter in a packaging cell line. Now into such a cell line, you have now introduced the same LTRs, but instead of GAG Pol envelope, you have your gene of interest, which contains a psi signal. Now by a single recombination, since both these genomes are in the same cell and there is a homology between the LTRs, by a single recombination event you can actually generate a wild type virus. Although recombination is a rare event, among let us say a million viral particles that is coming out of these packaging cells, 1 or 2 may be a wild type virus. So there is a chance that by a single recombination event you can actually generate a wild type retrovirus instead of a recombinant replication defective retrovirus. This is very, very dangerous, especially if you want to use this kind of recombinant retroviruses for human gene therapy applications, which we will discuss in the next class. So to prevent this kind of a problem, these kinds of cell lines in which all the viral proteins are being expressed from a single construct are known as the unigenomic packaging cell lines. From this, people went on to developing what are called as the bipartite packaging systems, wherein you produce all the viral proteins not on one single vector but on 2 different vectors. For example, in this case the GAG and POL envelope is expressed from one particular construct and the envelope is being produced from another construct. So you have a packaging cell line in which GAG and POL is being produced by one viral LTR, envelope is produced by another LTR, and if you now introduced your construct recombinant retroviral vector into such a packaging cell line, at least 2 recombination events are required for generation of a wild type virus, which is much more rare than a single recombination events. So to develop much more safer vectors, people actually started replacing these viral NTRs expressing this with mammalian promoters such as CMV and so on and so forth, so that there is no homology between the promoter regions and recombination will not be possible. So a number of safeguards have been developed in generating these kinds of recombinant replication defect retroviruses, and using these kinds of safe features, first generation these unigenomic packaging cell lines are called the first generation retroviral vectors, the bipartite is called as the second generation, and the third generation much more modifications were made into the packaging cell lines. Like I said, you replace a viral LTR with that of a mammalian promoter and so on and so forth. So a number of safety features have been incorporated so that the chances of a wild type retrovirus emerging out of this packaging cell lines is very, very, very minimal. So they become very safe, and such viruses can now be used not only for expressing genes in mammalian cells and culture, but also can be used successfully for human gene therapy. So as I said, multiple recombinant events are required for the emergence of wild type virus when you use these bipartite vector systems. In the first case, the vector containing gene of interest has to now recombine with this particular construct, so that you will now have a viral genome expressing only GAG and POL. Now that alone is not enough for emergence of virus. Then again this has to now recombine with a construct containing the envelope protein. Then only you will get all the 3 viral proteins in a single viral genome, and then only the chance of a wild type virus emerging out is possible. So by using these kinds of a modified packaging cell lines, the chances of a wild type retrovirus emerging out of cell lines was thoroughly minimized. Another important advance that took place in the area of developing recombinant retroviral expression systems is altering the tropism of the virus. I told you the envelope gene of the retrovirus determines what kind of a cell line it goes and infects. So if you can alter, suppose you have a packaging cell line which actually producing one kind of an envelope protein. Now instead of producing the envelope protein, if you now produce a different envelope protein, and the virus that is emerging out of that particular packaging cell line will have a different tropism. So by changing the envelope protein which is being expressed in a packaging cell line, you can actually produce viruses which have different tropisms. So based on their tropisms, the viral envelopes are classified according to the receptors used to enter the host cells. Ecotropic viruses can recognize a receptor found only on mouse and rat cells. That means these ecotropic retroviruses can only recognize murine cells, either mouse or rat. They cannot recognize human cells, so they will not infect human cells. Whereas amphotropic viruses, they recognize the receptors found on both mammalian cells, that is both murine as well as human cells, so they can infect both human as well as murine cells. Dual tropic viruses recognizes two different receptors found on a broad range of mammalian cell types. A pantropic packaging cell line, this is very important. This pantropic packaging cell line actually produces a virus that can infect both mammalian and non-mammalian cells, and using such cell lines, virions are pseudotyped with the envelope glycoprotein from vesicular stomatitis virus or VSV glycoprotein, which mediates viral entry through lipid binding and plasma membrane fusion. So you have now a packaging cell line which is now producing an envelope protein of a different virus, namely the vesicular stomatitis virus. So when you now introduce a recombinant retroviral vector into such packaging cells, the recombinant virus that is coming out will contain the VSV glycoprotein, and this VSV glycoprotein now can go and infect a broad range of host cells. So I actually altered the tropism of a retrovirus. These are actually called as pseudotyped vectors. So step, I think we will not go into the details. A number of modifications were made to develop packaging cell lines expressing the VSV glycoprotein gene. So a number of packaging cell lines have been expressed, have been developed, each packaging cell line expressing different kinds of envelopes. So depending upon what kind of envelope is being expressed by that particular cell line, you can either generate a dual tropic virus, amphotropic virus, ecotropic virus, a pantropic virus. The differences between these tropisms are actually listed here. Like for example, ecotropic viruses can only infect the murine cells, they cannot infect human cells, whereas the amphotropic viruses can infect both murine as well as human cells, and so on and so forth. Again, just like I told you for the adenoviral vector systems or adenovirus expression systems, a number of companies are also now selling these retroviral packaging cell lines as well as retroviral vectors. So if you want to now express your gene of interest using a recombinant retrovirus, all that you have to do is to go to companies such as CloneTech, buy an appropriate retroviral vector and buy an appropriate packaging cell line, clone your gene into this retroviral vector and generate a recombinant retrovirus of your choice. You can either generate, if you use an EcoPack 2293 cell line, you can generate a ecotropic retrovirus. If you use an AmphoPack cell line, you can use an amphotropic retrovirus cell line, and if you use this particular cell line, you can use a dual tropic, and so on and so forth. So you don't have to develop these cell lines, they are all commercially available. All that you have to do is to buy the right kind of systems that you want and generate a recombinant virus of your choice. You can also, there are also a number of laboratories across globe who actually developed a number of these retroviral vector systems. For example, Nolan's lab in Stanford University have actually developed a number of retroviral vectors. For example, here you have a retroviral vector containing the 5' LTR and 3' LTR with a packaging signal expressing the LAG-Z beta-galactosidase gene, and here it not only expresses the beta-galactose gene, it also has an internal ribosome entry site containing a neomycin marker. So you can actually select cells expressing the LAG-Z gene by neomycin resistance, and you also have vectors, for example, where you have a green fluorescent protein expressing from the internal ribosome entry site, and so you can actually these retroviral transfected cells will actually can fluoresce because they are expressing GFP. So a number of such retroviral vector systems either are commercially available from companies, or you can actually buy from some of the investigators who actually construct this kind of a retroviral expression vectors. Some of the major limitations of the retroviral expression systems are their inability to infect non-dividing cells. Retroviruses cannot infect non-dividing cells, and another biggest problem is they randomly integrate into the host chromosome. They can go and integrate anywhere in the host chromosome, and when that happens, when it goes and integrates either an oncogene or a tumor suppressor gene, it can lead to cancer. So there are problems of retroviruses using, especially in the case of human gene therapy. So to overcome some of the problems associated with the retroviral vector systems, the lentiviral vector expression systems were actually developed. One of the major advantages of the lentiviruses is that they are also retroviruses, but they can also infect non-dividing cells. That's one of the major advantages of a lentivirus expression system. So the lentiviruses are much more safer than the retroviruses. Now let's try to understand how lentiviruses can be used as gene transfer vehicles. The lentiviruses belong to the retrovirus family, but they can infect both dividing and non-dividing cells. They are much more complicated in their organization because they contain 6 additional proteins, namely Rev, VPR, VPU, NEF and VIF. I have shown here the difference between a retrovirus. As I said, retrovirus contains only 3 genes, GAG, POL and envelope, but lentiviruses, in addition to GAG, POL and envelope, contains a number of other accessory genes. The best studied lentivirus is the HIV, or the human immunodeficiency virus. So again, using the same strategy, how can you generate a lentiviral recombinant replication defect lentivirus? Again, you generate a lentiviral vector where you delete certain viral genes and introduce your transgene, and then you generate a packaging cells. Now what is the difference between a lentiviral packaging cell line and a retroviral packaging cell line? In a retroviral packaging cell line, you have to express only 3 proteins, GAG, POL and envelope. Whereas in the case of a lentiviral packaging cell line, you also have to express these additional proteins which are essential for the successful outcome of a lentivirus. So all the other lentiviral proteins are expressed by these packaging cell lines. Usually all of them are not expressed in the same construct, again to avoid the generation of the wild type virus coming out. So they are expressed in 2 or 3 different constructs, so that the chances of a wild type virus emerging out is minimized. Again, just like the retroviral vector systems, a number of efforts have gone to developing very safe lentiviral expression systems. You have what is called the first generation vectors, where there is limited homology between vector and helper sequences, separation of the helper plasmid. Still accessory HIV accessory regions were still retained in one single packaging plasmid. So the chance of a wild type HIV emerging was very high in the first generation vectors. In the second generation vectors, the elimination of accessory genes from the packaging plasmid, there is no effect on the virus vector titer. They retain the property of transduction of many dividing and non-dividing cells. They are much more safer compared to the first generation vectors. The third generation vectors are actually called as the self-inactivating vectors or SIN vectors. They had a deletion in the enhancer region of the 3' UTR that results in a transcription inactive vector that cannot be converted into a full length RNA. So the chance of wild type virus emerging out of such packaging cells is completely minimized. So replication compliant viruses cannot come out of such packaging cell lines. They hampers the mobilization by wild type HIV, may reduce the risk of tumorigenesis via promoter insertion. So just like the retroviral cell lines, the number of developments took place to develop safe lentiviral packaging cell lines, so that safe lentiviruses can be produced from these packaging cells. Again, just like adenoviral expression systems and retroviral expression systems, number of commercial lentivirus expression systems are available. I have now given here. Again, CloneTech actually sells many of these lentivirus expression systems. Like the you can actually generate a recombinant lentivirus which can express, which can infect either mammalian, either murine cells or human cells, because they contain the VSP glycoprotein as the envelope protein. Or you can also generate recombinant retroviruses which are ecotropic. That means they can infect only mouse or rat cells. So you can decide if you want to express your protein of your interest in a mouse cell line, you generate an ecotropic cell line. If you want to express your protein of interest both in mouse as well as humans, you generate a amphitropic cell line using the appropriate packaging cells. The complete details are available if you go to the company website. It actually tells you how to clone your gene of your interest and how to transfect these packaging cells so that you can generate the recombinant lentivirus. All details manuals are available. All that you have to do is to clone your gene into a vector system and follow the protocol, and you can generate a recombinant lentivirus of your choice. The recombinant lentivirus represent the latest generation of powerful multi-purpose vectors for delivering genes into almost any mammalian cell types, including primary cultures, non-dividing cells and stem cells. Among all the expression systems we have studied so far, the lentivirus have become the most popular and most widely used mammalian expression systems as I speak today. And they are the most efficient of all the types that we have studied so far. Again, if you go to the website, a number of modified versions of these lentiviral expression systems are available in many of these companies. You can either express your gene constitutively, you can either express your genes in an inducible manner using appropriate inducers like tetracycline and so on and so forth. You can have fluorescent tags attached to your transgenes so that the cells which are expressing your genes will flourish because you are putting GFP, and you can also select for puromycin and so on and so forth. Bicystonic expression vectors. A wide variety of lentiviral expression vectors are available from companies such as CloneTech. You can just buy and then do whatever you want. this is just another system that is sold by CloneTech, actually shows how you can regulate your expression. Once you generate this kind of lentiviral vectors, you can actually regulate the expression of your transgene by using molecules such as tetracycline. I will not go into the details of the system. One can just read and go through, or go to the company website and look at it. These are all called as inducible lentiviral systems, where the transgene expression can be induced by molecules such as tetracycline, because your gene is cloned under the tetracycline regulatable promoter. There are also compounds like retronectin, which enhances the efficiency of transfection. So the retroviral vector, the retrovirus vector can efficiently infect the cells, so that you get very high efficiency of transfection of a mammalian cells. So the retrovirus binds to a viral receptor, enters, but if you have compounds like retronectin, usually many of these mammalian cells also have receptors for certain regions of this retrovectin as well. So not only the virus will enter not only through the viral receptor, but since these viruses bind to this retronectin, by interacting with multiple receptors, efficiency of virus transfection is enhanced. So a large percentage with retroviruses if you include compounds such as retronectin. So the efficiency of gene transfection is thoroughly increased by using compounds like retronectin, again available from companies like CloneTech. So I just given some of the very informative brochures and articles that are available for you to study further. CloneTech again gives a very detailed manual on how you can use various lentiviral expressions available with them, how one goes about constructing these lentiviral vectors and so on. Similarly, they also sell very interesting and very informative manual on retroviral gene transfer and expression. One can read up a little bit more and try to understand how various retroviral vectors are being generated. There is also a very informative article in Annual Review of Biochemistry, Gene Therapy 21st Century Medicine, where it discussed the various viral vectors that are being generated and how these viral vectors can actually be used for gene therapy, which is what we are going to discuss in the next class. I think I'll stop here. Thank you.